So hello everybody, I'm Fani Mantzouridou, I'm coming from Greece, and the project that I will present today is the FAIRferm project. The FAIRferm project focuses on the verification of microbiome and metabolome time series datasets from food fermentation processes. The consortium that supports this project consists of 6 ELIXIR nodes, EBI, along with the food and nutrition and microbiome community. To set the stage, fermented foods are products that derive from controlled microbial growth, and the food composition is due to complex enzymatic actions. In the community of fermented foods, the plant-based fermented foods are very important in terms of nutrition and are very critical for the sustainability and security of foods. However, the management of the fermentation process to produce plant-based products is based mainly on empirical knowledge, and this results in the fact that we don't know the dynamics of microbial communities that shape the fermentation process, but also the metabolic network that is developed in order to have nutritious and safe food. And for us that we work on this field and produce many data, we really want to know how we can harmonize all these data sets in order to be able to increase knowledge and standardize the processes. We know very well that fair storage of data so that everybody can reuse them and know where to find and reuse them is very important. Also, it is very important to create common open databases to reduce the database, the data that are produced in silos, and the researchers to be able to provide and to share data in systematic approach. The big problem is also the fact that there is a poor documentation of all these data processes. Okay, and for this we need to know the minimal required metadata that is needed in order all of these metadata to be linked and be able the datasets to talk to each other. And of course, advanced usage and current development of current use and development of sophisticated bioinformatic tools in order to understand the relationships between the microorganisms that drive the process is very important. We know all this, so we have set up the project vision, which is to improve metadata standards, interoperability, and fair pipelines for microbiome metabolome time series datasets from plant-based food fermentation. We have 6 different activities, 5 deliverables and 3 milestones. We are very happy to complete the first deliverable, where we try to find the gaps that exist in the ELIXIR resources that can be used as a tool to repository our data. And finally, the predicted outcomes of the end project is to have harmonized metadata standards for multi-omics time series datasets, to have FAIRified datasets using ENA, MGnify, and MetaboLights, and link all these via the BioSamples ID. And of course, to be able to develop fair pipelines for temporal analysis of our datasets. The training is running, and of course, we will be able to produce related training material and standard operating procedure material for all the researchers that want to follow this. Thank you all.